So Dolph, could you tell us something about what is so special about stickleback species pairs that are found up on the Sunshine Coast?
I have been working on them since the late 1980s.
Right.
So I'm a professor here at UBC, and I study how new species form and become different from one another.
Yeah.
And at that time I was looking for a system that would allow me to investigate how new species formed, and I learned about these stickleback species pairs. And one of the several reasons sort of drew my attention to them. One is that they are among the youngest species on Earth in anything, any organism.
Right.
And we know that because the lakes in which they occur are only something like 10,000 years old.
Yeah.
Another thing that made them special and attractive for study was that there's more than one species pair. It looks like pairs of species have evolved over and over again.
Yeah.
And the ability to predict anything in evolutionary biology is rare, and the repetition of events in Earth's history are special because they tell us that sort of general laws are at work.
Right. Yeah.
And I was very interested especially in the role that environments play in the origin of species. Right. And another reason that we were interested in them is because the species within a lake, like Paxton Lake, there are two forms. One of them we call the benthic, the other limnetic. They're so extraordinarily different from one another in the way they use the environment.
Yeah.
And so this allowed us to address what was, up until recently, a sort of burning question in evolutionary biology, which was whether adaptation to environments had anything to do with the origin of new species, as Darwin supposed, or whether it was primarily chance factors in history and the genetic changes that occurred by chance that was sort of behind the origin of species. And it was the repetition of these species pairs, the fact that they had occurred over and over again, that allowed us to solve that problem.
Right.
So we didn't start out working on these species because they were endangered, but it was partly because of the extraordinary scientific value that they possessed that others began to learn of them, and we became sort of conscious ourselves that they were pretty special, and also that many of the reasons that we found them so interesting scientifically were also some of the reasons why they are so vulnerable.
Yeah.
So being very young, for example, means they're very prone to collapse when environments change, and that's exactly what happened already in Enos Lake.
And there are people all over the world studying these sticklebacks from the Sunshine Coast.
So these are certainly some of the most interesting stickleback, and all around the world people are working on stickleback and have for some time. Nico Tinbergen won the Nobel Prize for his work on the behavior of stickleback and other organisms.
It's a post office fan.
But as a result of collaborations that people here at UBC, my group, have with other people working at Stanford University in California and at the Max Planck Institute in Germany, and also at the University of Bern in Switzerland, that we have people doing a lot of work on the stickleback species pairs. And a lot of that work is focused especially on understanding the genes that make them different.
Right. And you go up to like these lakes on Texado Island and on Nelson Island, and you collect specimens and you do work up there and bring stuff back here to UBC to study.
We do, yeah. And so in the old days, when we started work on the system, they weren't endangered, and I should say they weren't listed as endangered, and so there were very few restrictions on carrying out work in the lake. So we built enclosures and we did sort of transplant experiments. We tried to ask, how much better is the benthic in the benthic environment compared to the limnetic, to try to gain some initial understanding about just how important the differences that they have were to their abilities to thrive in the environments. But as they now have become federally listed, it's actually more and more difficult to do
Right.
work in the
yes, that's,
lakes,
that's
and
typical.
that's a good thing. I mean,
Yeah.
it was partly our work that drew attention to them, and that made others realize just how special they were. So as a result, a lot of the work that we do on the stickleback now is carried out at UBC.
Yeah.
And so each year we are allowed to go and collect a few and bring them back, particularly if the work sheds light on their conservation status and on the prospects for maintenance and recovery.
Because I remember when I first met up with you, it was because we were worried about on Texada Island, the locals there didn't really know what was going on with the sticklebacks at all, that weren't really very aware of what was happening until we formed the Texada Stickleback Group. But
That's right, your work was actually really important in getting people to recognize what was basically in our backyard.
Yeah. And what we really want to do now in our new facility, in the PODS facility, is to show it off even more about how important this is by having a big exhibit about the stickleback species pairs. And,
I think an exhibit in PODS would be an absolutely fantastic way to make people aware of the species that occur locally and why they're interesting scientifically, why they're important biologically, and why they're threatened and what we can do about that.
Brilliant. And we're also building laboratory facilities for marine and freshwater students and researchers to come and be able to work on the spot.
That would be a marvelous facility.
So we think that would be valuable.
I think that would be a great advantage for, especially because, in moving fish around, it can be difficult to get them here all in one piece, and because we're allowed to collect so few, if a few fish die, tough luck, we can't go back.
So Dolph, you approve of PODS then?
I do. I look forward to seeing it. I think it has every chance of being of great value, educational value in particular. Letting people know what's in their backyard, why it's so important, why the species are vulnerable. But I believe it would also be useful to us scientifically, especially when we are transporting individual animals from the lakes back to UBC to have a sort of a mid-station. I think would actually greatly improve our chances of bringing back the fish alive and in good shape.
And do you feel that there's still a lot of research that needs to be done out there? I mean, I know you're doing a huge amount on sticklebacks, but it seems to us there's a huge gap in particularly marine research in that area. I mean, I don't know how do you feel about that? Is it?
Yeah, I think, so I do think there is a lot of work still to be done on the stickleback species pairs because, it's only recently that we've discovered some, very important things about the species pairs. We, until our collaborators began to discover just how many genetic differences there are between the benthic and the limnetic form, we had no idea. It's only in the last couple of years that we have been able to say how many individuals in the lake are actually hybrids. And so we're in a better position now to look at hybridization, but also to try to put the whole picture together to understand how these species actually persist in nature, particularly when they're so young, particularly when they're prone to hybridizing.
Just one last question. The last time we met, you said to me about in the introduction of the Origin of Species, there's a phrase where he calls natural selection, Darwin calls natural selection the mystery of mysteries, and you said to me that you would have solved that within 4 years. I just wondered how close you are now.
Yeah, that was a little optimistic. So at that time I was mostly interested in the role that natural selection and adaptation to the environment played in the origin of species. Right. And when the work on the species pairs began, we knew almost nothing about that.
Yeah.
And the stickleback species pairs have played a large role in changing our understanding and increasing our knowledge about the role that environments play in the very origin of species, not just in their maintenance.
Right.
And but now that we have this sort of general framework in mind for how species originate, stickleback species, but species in general.
Yeah.
We now have more questions about, well, just exactly how did this happen? How exactly did natural selection, acting on traits like jaw bones and muscle mass, cause the genetic changes that now form the barriers to interbreeding between the species. So that level of detail is still missing in general, not just in stickleback species pairs. And now that we know so much about the genetics of stickleback, I think we have a good chance of making progress on that front as well.
Right.
You asked about marine stickleback, though, and I'm becoming much more interested in marine stickleback.
Oh, good.
And for two reasons. I actually think that stickleback, because they're so abundant, because they have such a large distribution that ranges from Alaska down to southern California, I actually think that stickleback will serve as a model for understanding a project that we call evolutionary rescue. Basically how adaptation occurs in the face of environmental change.
Wow.
And as far as marine fishes go, stickleback have a pretty short generation time.
Right. Yeah.
And there's the prospect of actually understanding how local populations are changing and will change over the next 50 years.
Fantastic.
So I'm applying for funding to take samples of stickleback right from Southern California all the way to Alaska. I'd like to maybe sample 50 individuals from 10 different marine populations across that span. And part of the goal of that study is also to identify genes that are changing in frequency, possibly as a result of adaptation to changes in climate and pH. And then to repeat this for salmon species that have a similar latitudinal range. So we're going to try and do this for Chinook salmon at the same time.
Oh wow!
Which have the same latitudinal range. So they also occur from California to Alaska. And so work that we've reviewed, but that has gone on in labs around the world, has shown that often when species adapt to the same environmental changes, they use many of the same genes. And so we think, and there's a difference between stickleback and salmon in how easy it is to measure genetic variation. So the sequence of stickleback genome costs $240 now, whereas a salmon is 10 times that amount.
Oh, right.
So we think that actually stickleback will be useful for identifying genes that might be important for adaptation in marine fishes in general. So that's a project we're just starting out.
Well,
so Pender Harbor would be an obvious target.
That's right.